All right, welcome everybody to our webinar on analyzing the UK Biobank dataset with RStudio Workbench. We know you'll have questions during the webinar. The best way to get them answered is to post in the Q&A, which you'll see in your Zoom toolbar there, where our speakers and panelists can see and answer your questions accordingly. We have a few members of the DNAnexus team on, Phil Sweeney and Chai Fumtumasan, who will be helping answer these questions throughout. And we'll also have a dedicated Q&A section at the end of the webinar. You can also take a look in chat where there'll be helpful links and info that you can access as well. These slides and recordings will be available within a day or two by email and on the DNAnexus community, which I'll mention in a second. You can go there, be able to find them. There's a dedicated post, and they'll also be in your inbox within that time frame. So as I mentioned, the DNAnexus community, this is an online community forum that is moderated by both DNAnexus and UK Biobank teams. You can jump in there, ask questions about the research analysis platform. You can also find news, events, announcements, signups for user workshops, upcoming webinars. The forum is free to join and registration takes less than 2 minutes. And if you are an early career researcher or a researcher from a developing country, you might actually be eligible for the UK Biobank Platform Credits Program. This is designed to allow researchers to explore the research analysis platform in detail, develop and test tools and methods, and undertake analysis to support their research project. So you can visit the website today. You can scan that QR code, or like I said, slides will be passed around afterwards, and I'll post a link to it in the chat for you to be able to access that. And then if you are an approved researcher and are not already on, you can register for the research analysis platform. This is available for all UK Biobank researchers to access. It allows you to access and analyze the UK Biobank dataset securely in the cloud and utilize the tools mentioned in our webinars, like this one. It's the data analytical tools in cloud, all your fingertips. And one note about the RStudio Workbench that we'll be talking about in this webinar. It is available free of charge for all wrap users now until August 31st. Standard platform pricing for computing, storage, and egress rates apply. And you can find more information for pricing via documentation listed on this slide and that I'll post in the chat. So now I will introduce our speakers for today. First is Alex Stempar, who's a bioinformatician at UK Biobank. Alex is an experienced computational biologist working in genetics, genomics, statistics, machine learning, and data visualization. His current work is focused on the research analysis platform. We also have Phil Bauscher, who's the Director of Healthcare and Life Sciences at RStudio. Phil is a published author and award-winning speaker, having given over 100 R talks and workshops in 4 countries to an estimated 20,000 people. His work focuses on innovation in the pharmaceutical industry with an emphasis on interactive web applications, reproducible research, and open source education. And then we have Anastasia Sedlikova, a principal scientist at DNAnexus. She's an experienced bioinformatician who also has considerable experience in data science work for academia, pharma companies, as well as financial companies. For the last 10 years, Anastasia has been teaching adult statistics, programming, and HPC cloud technologies, and continues this work here with workshops and webinars for the research analysis platform. You may recognize her if you've attended some of our other webinars, like our overview webinar or recent RStudio training one that we had last month. So without further ado, I will pass it over to Alex to kick us off.
Hello, thank you very much for a very nice introduction. And I would like to welcome you all from the UK Biobank headquarters in Manchester. And in today's talk, I would like to show you 2 things. So first of all, I would like to do a quick introduction to the Biobank mission and the dataset. And the second thing, I would like to show you a live demo how to use the platform and what we are using the platform, or I'm personally using the platform in Biobank for. So to start, in UK Biobank, UK Biobank is the huge study that started with gathering 500,000 volunteers, age 40 to 69, most of them from England, some from Scotland and Wales. And they were the samples from them, plus the intensive baseline interviews, were taken from the 22 assessment centers. And then we also can look at our participants' health development and image and re-image their participants. So at the initial stage, 20,000 of participants have been reassessed. And then this is the effort that is ongoing right now. 50,000 participants were re-invited for quite extensive body scanning with different scanning techniques. And the target is to have 100,000 participants scanned, and some of them done with the multiple scans. So talking about the data. So the first component of the data is the very rich, very big phenotypic data set. So it's a table with more than 20,000 columns that incorporate all of the baseline data collected with the interview. And also our participants gave us an access to their health records. So basically both the hospital health records and the primary care GP health records. And in Biobank, we like to set that we are 10 years ahead, and we always will be. And this figure quite nicely show why this is the true. So our participants were recruited when they were generally healthy people. And as their age, there is more and more occurrences of the diseases happening. So the older the data set become, as the time progresses, the more and more participants would be showing the different kinds of the diseases. And it allows us to do this very in-depth studies, which basically having a sample population, matching sample population of the healthy people, and having the very extensive data about their lifestyle, about their other medical history, and seeing how the disease is formed on the basis of all of those factors. So as you can see in 2020, there is the numbers of the observed diseases like diabetes, stroke, depression, and so on. And this is the predicted numbers. So as the time progresses, we will be observing more and more participants developing these diseases, and we can look at all of this underlying information and try to understand why it's happening. So we've got quite a lot of modalities of data. There are, for example, like wearables data, but the two most important modalities are the omics data and imaging data. So from the omics data, we have for pretty much all of the participants, we've got a microRNA and imputation. So microRNA was genotyping around million variants, and then we imputed more than 90 million variants from the population. Then we are finishing right now the exome sequencing. Last year, we released almost whole sample already. This is approximately 2% of the genome that is being sequenced, and we identify tens of millions of the variants in this population. And finally, the effort that we are doing right now is releasing the whole genome sequencing for all of the participants. So this uncovered billions of the variants pretty much in all of the places of the genome for participants. And by 2023, we plan to release all 500,000 participants being fully whole genome sequencing. In addition of that, we've got the MNR metabolomics data. We've got the proteomics data, which would be released starting 2023. And we've got all of this rich imaging data, that examples you can see on the right side. So there is a question, because the biobank is already operating for quite a lot of time. The question is why we need the research analysis platform, why we need this cloud environment to analyze our data. And there are two components to that. So the first component is ever-growing data set. So especially the whole genome basically push this data set to the limits. But if you take all of the imaging, exome sequencing, microRNA imputation, we've got more than 21 petabytes of the data. So this is kind of the mass of the data that is not trivial to move around, even if you have the best-in-class compute and network access infrastructure, and becomes prohibitive for most of the research centers to store this data in the one place. And the second reason is the increasing number of the researchers in the biobank and increasing number of the researchers from all over the world. So as the mission of the biobank, we want to democratize access to the research. So not only researchers from the rich institutes in the rich countries would have access to this data, but pretty much everybody from the world who can apply would have an access to this data and can analyze the data in the cloud. So this graphs nicely show the number of the publications using the biobank datasets. As you can see, they are growing quite fast. and also the composition of the researchers that are registered with the biobank. So as you can see, in 2016, they were pretty much coming 50-50 from the UK and the rest of the world, and basically mostly coming from the US and from the UK and the US. And right now we've got the researchers registered from whole world. So we think what we are doing is really working and helping to access this great data set. So with the RAP, there is quite many ways to access the RAP and basically like interact with the data. And obviously our today's seminar is focused on the cloud workspaces, and in particular using the RStudio in cloud. So the cloud allows to do this quite a lot of things, like running RStudio, running all of the supported languages like R, Bash, Python, Stata. Obviously, RStudio is focusing mostly on R, but it allows to interact with all of these other languages. There is the integrated snapshot and session management. You can basically make the snapshots of the session. You can save the data in your projects. You can access Spark infrastructure. You can access the machine learning instances with the GPU enabled, if you are doing something like a deep learning or GPU-assisted workflows. And also distributed computing, like if you are working with the Pictionomics dataset, the Hail is great framework working at the top of the Spark. And the power of this is that you can access this resource in the cloud, and with just a few lines of code, you can get the results. So you can just grab the phenotypic data as shown in this first slide. Then you can mix and match the phenotypic data. So for example, this slide is showing the number of participants that were showing some kind of indication of the hypertension. And we can use the self-reported hypertension that participants reported during the initial interview. We can use the extract from the medical records, and we can use the blood pressure cutoffs. And as you can see, we can mix and match those to make our cohort best suited for the research questions we want to ask. So then we can access all of the genetics data. So we can do all of the stuff like basically seeing the population, right, doing the PCAs and calculating the covariates for the population structure. And then we can do the stuff like GVAS, for example, and this is quick plots showing how the GVAS may look like. Then we can extract the interesting variants and, for example, do the interactive annotation. And all of this can be done literally in minutes with a few lines of the code in the cloud. And this is a quick, basically, this is the quick slide showing the resources. So if you want to access the data, or if you want to have some help with supporting, you can contact either DNAnexus or our support at Access Biobank AC UK, and we are very happy to help with everything. And before I move to the live presentation, I would like to acknowledge, first of all and foremost, all of our participants who generously donated their data and their samples to this amazing project. Also, all of our funders, which is basically Medical Research Council, Wellcome Trust, the Department of Health and Social Care, British Heart Foundation, and Cancer Research, for generously supporting us and supporting this effort. And all of my colleagues and consortium members that make all of this project possible. So right now, moving to the second part of the presentation, which would be a live demo. So I hope you can see right now the browser window. So for those of you who are familiar with the RStudio, it looks exactly the same in the cloud as would look in your desktop, with just a few little cloud-specific controls being here that are very easy to find. But if you are using RStudio in the desktop, you would find yourself here at home very easily. And before we were giving the access already to all of these resources using the Jupyter Lab, which is quite nice for writing the notebooks. But RStudio gives you like a second level of things that you can do, because you can develop the web applications, you can develop packages, you can do much more involved development that is hard or impossible to do in the simple notebooks. And also you can use RMD notebooks, which are much better for like generating reports and basically code management. So this is the two great things that you can do with the RStudio, and the two great things I would like to show you right now. So the first thing, the problem that we have with analyzing our data is that we have this huge cohorts of the data that pretty much like there are no tools for visualizing the stats and visualizing the QC parameters for those. So one of the things that we need to do in Biobank, for example, to visualize the stats on 500,000 VCF files, is to run the tools that would help us to fetch and visualize those stats. So what I'm showing you right now is basically the code for the Shiny application. And you can see there is a standard code for the server and standard code for the UI. So this is the same thing as you would develop in your desktop probably. And then if you just click the Run app, you've got all of this running already on the cloud. And then as you can see, there is like a 20,000, this is the stats on the 20,000 VCF files. So you can see numerous stats like a number of the records, number of the SNPs, transitions and transversion ratios, and so on. They are really helping us with doing the QC and spotting the samples that might be problematic. And the very nice thing about that we can do here is basically this live visualization. So we can, for example, click on one of the samples and see the plots representing the its coverage over the threshold of 30. Or you can see the substitution right now. So this is the plot of the substitution, and this is the transition versus transversion ratio based on the quality score. And then you obviously have all of this interactivity that Shiny apps come from, so you can change lots of the parameters here if you want to make a visualization, visualizing the thing that you are mostly interested in. You can see all of these quality plots. You can see the tables with the actual quality stats. And you can also see the summary stats. So this is, for example, the number of the records. And then this dot represents the sample that was basically highlighted. And now, if you can see it, so basically like going, for example, for the coverage, we can quickly iterate to the samples and just one simple click, we are seeing the next sample. So that's the power of using the RStudio and cloud developing this application, that you can develop it very easily. And when the application is ready, you can basically share it with your colleagues using the applets that are developed, the technology developed by the DNAnexus. And the last thing I would like to quickly show you here, and I believe the speaker that follows will tell you much more about it, is RMD notebooks. So this is like very nice, very intuitive thing to write the code and then write basically also some annotations to that in the Markdown, and then seeing all of the plots here, which is like very nice for doing the different manipulations of the data and looking at the data. But what I most really like about it is that you can click this button that is clicking it, and you can produce a very nice PDF reports. So as you can see, this is the PDF report that reports all of the stats about the quality of our cohorts. We're breaking up on the different cohorts and breaking up on the different sequencing provider centers. And thanks to it, when we are receiving the new samples, I don't have to go through writing this report again. I can basically reuse the code and reuse the previous annotations basically to generate the new report on the new sample that is coming in. So I hope that it was somehow useful, and it showed you it's like a little bit recaptured or recap to what we are doing in the Biobank, show you what we can do in the cloud. So thank you very much for your attention.
Fantastic. Well, thank you. Let me go ahead and share my screen here and welcome everybody. So today, what I want to take you through is take a few steps back. And for those of you that are new to RStudio and this type of analysis, help you get started and see how to build an end-to-end workflow like you just saw. But really, how do you do it from the start? And so when I speak about workflows, what I'm talking about for today is how do you import some data, clean it up, visualize it, and then communicate those results. And so this is typically referred to as the data science workflow, but it could be the research workflow, the drug development workflow, all of it's very, very similar. And so what I'm going to show today is how can you do this within the DNX platform, and how can you do this on the Biobank data right inside of RStudio. And so for those of you that aren't familiar with RStudio is an IDE, and that stands for Integrated Development Environment. It's basically the place where you go and you do your analysis and you do your work and build those workflows that you're going to use on the genomic data that you have. And so this would be your first introduction here to RStudio. You can see that I have a lot of projects and a lot of sessions going. And so this is where I would first kick off the work. But because today we want to make sure we're focused on reproducible research, there's a couple things that I would recommend that you start with right off the bat. So the first thing is I wrote a paper last year focused on the 3 tools for reproducibility in the research or pharmaceutical space. And a couple things that we talk about in this paper are RStudio projects, the RINF package, as well as using version control. And so I'll make this paper and the other examples that I build throughout today available to all of you inside of a repository that I'm going to start and build. So I'm first thing right off the bat, when you first go to do this analysis, is you want to make sure you have good, clean record of the code that you've developed. So the first thing I'm going to do for today is I'm going to launch a repository. And so I'll just call this UK Biobank. And so now that I have my repository made, I can go ahead and add a README file to this and create the repository. And when I do this, it's going to set up an environment now on GitHub where I can store my code and my examples. And so now that this has been established, I can take this repository, I'm going to go back into RStudio and I'm going to launch a session. And let's go ahead and call this webinar, which is going to be the session that we're working on for today. I'm going to launch this session and RStudio is going to kick me into that environment where I'm going to do my analysis for today, which is the webinar. And so right off the bat, it's going to take me into an RStudio session. Go ahead and launch this. And so what we're going to start with is we're going to go from here and we're going to launch an RStudio project. And so I'm going to click on new project, version control, Git, and I'm going to put inside of here the repository that I just made. And so now I'm going to create a link to the UK Biobank repository that I just made. And I'm also going to establish that link inside of RStudio. And this is going to make it so when I do my analysis, I'll have a place where I can publish and deploy code so that other people on my team can see that. And so right off the bat, the first thing that we want to think about for today is how do we get the data and do some of the analysis that we're going to do for today. So just give me 1 second. I'm going to import into my RStudio session the data for today. So for you, this might be a little bit different inside of the platform. And so this is what it would look like. And so I'm going to say, hey, let's go ahead and open up this file here, which is the data that we're going to look at for today. This is, I'm going to put my RStudio environment back and I'm going to go ahead and click OK. Now, while this is loading, what we're doing is bringing the local CSV data set that we're going to analyze in today's session directly inside of RStudio. And so I'm loading this from a local environment and I'm bringing it over into the cloud environment or into the server environment where we're going to do the analysis for today. And so this takes just a few seconds to get loaded. And once it's there, though, I've still got to convert it from a CSV data set over into an R data frame. And so we're going to go ahead and once this is loaded, have the data set so that we can start and kick off the analysis that we're going to do for today. All right, it's a little bit of a larger data set, so it takes a second to get loaded. It should be coming in. We'll just give it 2 seconds here to load while this is happening. All right. Right. And so once I have this data set loaded, a couple of things that we're going to look at for today. So I also wrote a paper about development and some of the key things that you'll see and hear about as you build your workflows is following what's called an agile workflow development. And so for today, what we're going to look at is how do we build an analysis inside of RStudio as a notebook. And then once we've developed that notebook and we've got it into a format that we like, how do we develop that into a dashboard? And then once we've got the dashboard working in the way that we like, how do we take that and convert it over into a Shiny app? And so these are the things that I'm going to show here over the next 15 or so minutes is how do you create a notebook of the workflow? How do you take that notebook and turn it into a dashboard? And then once you like your dashboard, how do you create a Shiny application like the one that you just saw in the previous section? So let's go back here and you can see that the data set has now been loaded into the environment, but we need to get it into the environment so that we can analyze these results. So to build this workflow, this reproducible workflow, what I'm going to start off with today is a notebook. And so my notebook is going to be what's part of the R Markdown ecosystem. And I'm going to build this workflow in this notebook to replicate what we saw as part of that data science workflow. And so the first thing I'm going to have are the packages that I'm going to use. Then I'm going to load the data here. And then once we've got the data loaded, let's go ahead and clean the data, maybe wrangle it up just a little bit. And then once we have the data, let's go ahead and create a visualization. And then once we have that visualization, let's convert it into an interactive visualization. And so this is going to be the workflow that we're going to build and analyze for today. Okay, so the first thing we need to do is let's get our data loaded into RStudio so we can start to analyze the results. So I'm going to click on the data and I'm going to import the data set. Now, when this happens, it's going to bring up a tool, and a lot of things I'm going to show today are really geared towards people that are new to RStudio, new to this environment. And so this is how you can get some of the examples quickly if you're new to doing some of the research in RStudio. So right off the bat, I'm going to get this bit of code here because this is going to import the data set that I want. And so you can see now I've got the data inside of RStudio. I've got 7,000 observations just about. And I'm going to go back to my notebook and I'm going to put that code in here for bringing in the results and the packages. And so hopefully you can see here now, I've got one package that I'm working with, readr, and we can go ahead and load the data in. Now, it's nice to be able to see the data inside of the notebook. So let's go ahead and put the name of the data set right there and run these results. And when we do this, what's nice about this notebook is it gives you this very interactive and clean experience where you can do your research. So you can now see, I can see my results here. I can always click on the data and pull up the results here if I prefer to see the data set a little bit bigger. So we're working through this workflow. We're getting to a point where we now have the data loaded, but let's go ahead and clean up some of this data. For that, let's go and use a nice tool that's inside of RStudio. And it's inside of RStudio because what we're going to build today for cleaning up the data and also for doing the data visualizations is a really cool tool called Esquisse. And it's basically a package that was developed by a team called Dreamers here. And it allows you to do some functionality inside of RStudio on data through a point and click tool. So it's great for beginners, and it's a really nice way to interact with your data. And so that's what we're going to do now. And when we go to work with this tool, it's going to bring it directly inside of RStudio through this add-in section here. And so I can scroll down and click on ggplot2 Builder. You'll see it right there. And when I click on this tool, it's going to launch the ggplot2 Builder tool. And when it does that, it's going to say, hey, which data set do you want to analyze? Well, we only have one data set for today so far, and that's the data set that we want to go ahead and start cleaning up. So I'm going to pick the data set that we've loaded. I'm going to import the results, but I'm not going to jump into doing the data visualizations just yet, because what I want to do is actually clean up some of the data. And so let's say just off the bat, let's go ahead and clean it up using the clinical significance variable here. And so let's say I want to get rid of a certain significance, and let's also get rid of conflicting significance here. And so when we do that, it's going to kick off the code, and you can see here the results for doing the data wrangling. And so I can close out of the tool, and now I can put that over into my next section for doing the data wrangling, where it's going to create a new data set here. Let's call this GWAS_clean. And I'm going to set it equal to the original data and then do the data wrangling. And when I do this, it's going to give me an error. And that error is because we need another package up here, which is part of an ecosystem called Tidyverse. And the package that we're going to use here for this is called dplyr. And so I just loaded the dplyr package into my section. And now let's go ahead and wrangle the results. You can see I just did that. Now I have a new data set here, GWAS clean, which is a subsetted data set of our original. And so now we can work on that clean data set and build some of those visualizations. And so let's go back to that tool and build the visualizations for today. And so I'm going to jump into ggplot2 Builder one more time. I'm going to load the clean data this time, and I'm going to import it into the tool. Now, this is a great tool for doing data visualizations, but there's other nice ones in the ecosystem. One of them that I've seen is Profiler 2. It's a great tool where you can also go and use R and build interactive visualizations. But for today, we're just going to use this as Squish tool, especially for beginners. It gives you a nice clean tool to use. Okay, so let's go ahead and drop in a few of these variables. So let's look first at the position. Then next, let's look at the log 10. And then for the sake of today, let's go ahead and group these by this section here. So we're going to look at the chromosome number, the position, and the log 10. And so this is the visualization that we just made. And so I can now click on this tool, get the code for this, and take it back into the visualization component. Now, when I do that, I'm going to run this. It's going to give me another error. That's because we need to add up here the ggplot2 package. So we've got this puppy loaded, and we now can load that package and come back down here and get the results. All right, so here's the visualization that we just made. Now, it would be really nice if this could be interactive. And so to do that, what we're going to do is take the exact same code that we just made. And this time, though, I'm going to create an object. So I'm going to call this data visualization 1. And I'm going to set it equal to the ggplot2 that I just built. And when I did that, you'll see now I have an object up here. And if I take dv1, it will just print out the exact same visualization, but I want to make it interactive. And so to do that, I'm going to kick over into a new function called ggplotly. And ggplotly is going to give me capabilities to make this interactive. Now I've got to load my package into here. And for this part, we're going to use a package called Plotly. And so I'm going to load Plotly and its functions into my environment. I'm going to scroll back down and then go ahead and hit play here. And now when we take a look at the visualization that hopefully will pop up, we should have now an interactive visualization. And we do, which is quite nifty. So I can actually now click on the charts and get an interactive visualization, which is pretty cool. Now, because we also created this notebook, I can save the file. And so I'll call this Phil's workflow. And once I save this, it gives me a notebook file. And so I can click on my files down here and see the research that I just made. Here's Phil's workflow. And then also, if I want to share the result with my colleagues, I can preview the analysis. And this will give me an output file, which is a notebook that I can share with others. I can put this on a web server. It's basically an HTML file. It's nice because people can come in here. They can download our markdown code from here. They can also see the results. They can see the data. They can interact with the data. And moreover, they can actually interactively engage with those visualizations that we've created. But this is pretty cool, and it's a nice thing. It's the workflow that we've made. But in that agile development, now maybe we're ready to start taking these and create a dashboard. And so for that, we're going to use a tool called Flex Dashboard, and this is our Markdown package for easily creating really nice looking dashboards using R. And so you can see a couple of the examples that I have here. And so I'm going to go back into RStudio, and this time I'm going to click on File, New File, but instead of going to a notebook, I'm going to go to an R Markdown script. And so the script I'm going to use here is a template, and so I'm going to go to Flex Dashboard and click Create. And now when I save this file, I'm going to call this one Phil's. Let's go ahead and call this Phil's Dashboard. And so I've got Phil's dashboard. I'm going to save it. Now, when I generate this report, though, you're basically just going to see a skeleton, not much here. It's just going to look like a nice, simple report. And so we've got chart A, we've got chart B, and we've got chart C. And so what we just did with our notebook, though, is now we can take these packages and this workflow and start to add these into the notebook. And so I'm going to simply add my packages over here. And I'm going to take the data wrangling component. I'm also going to include that at the top. where it's going to render and create my data sets. And so let's go ahead and do the data wrangling component that we want to do. And so once we've got that set up, we can now take the visualizations that we created. So we've got a ggplot2 visualization. So let's go ahead and put that into chart B. And then let's go ahead and take the interactive run, and we'll put this into chart A so it's nice and big inside of our visualization. Now we've got another section down here, chart C. And one thing that I've learned over my career is people like to see the data. And so let's go ahead and show people the data. And for that, we're going to use a package called DT. So let's go ahead and add the data tables package up here. Save this, and then we'll scroll and go ahead and add some sample code here so you can see it at the bottom in chart C. So chart C is going to be data, and the data that we want to load here is going to be the GWAS clean data set that we have. And so now that I've got my dashboard, let's go ahead and see what I've created now. There we go. And so it's rendering the report. Here's the HTML dashboard, and hopefully this puppy pops up. Give this 1 second. Here it is. And so just now I took the notebook, but now what I've done is I formatted it into an interactive dashboard. And so I've got the interactive plotly component over here. I've got the ggplot2 visualization that we created, as well as I've got the data. Now I'm a little bit zoomed in, but this is the data and the results. So I can click through the data and I can look through and see more data points as well. And so now we have an HTML dashboard that we've created and we can share it throughout the organization. But one thing that we might want to do is convert this into a Shiny application. You may have heard about Shiny, or you sit there and we just saw a Shiny application. And what's important about Shiny application is that we move away from the local HTML side, and we're going to move more into the server side. So it gives some really nice interactive components. And so Shiny is a popular tool for analyzing results. And so what we're going to do now is go ahead and do that. And so for that, I'm going to upload a Shiny application that I built called the Universal Data Visualization Shiny app. I'll include this in the repository that I'll share in the chat box here in a second. I'm going to go ahead and upload those results. And now when I've done that, it will bring the Shiny app into my project here. Here it is. And so I've got my universal app. Now there's a couple things that I want to change here. We want to bring into our application we want to bring in the data set that we're working with. So I'm going to go to my Shiny app and I'm going to read in here today the Biobank data, which for us is the GWAS clean that we want to include. And so I'm going to have GWAS clean. Now there's one last thing I need to do here. I need to convert the chromosome number over to a character. And so I'm going to add that section right here so that we have that functionality. And so now I can run this application. It should hopefully now bring up a Shiny app. And here is the Shiny app that we just made, and hopefully it works correctly. Looks like it didn't find the data, so let me go ahead and change this. UK webinar, UK Biobank, GWAS clean. Let me see. Should be in here, is it not? You know what?
You're missing the data folder. UK Biobank /data, then GWAS clean. Yeah.
Yeah, you know what? I think this was the problem right here. So we're going to go with this. Here we go. So we're going to actually work on the CSV data set and not the R data frame. And so let's go ahead and give this a try now and see if this works. I think it will. I think it will. And so here is we're starting from so what we're going to do, actually, every time the Shiny app runs up, it's going to convert that CSV data set over to an R data frame for this session of the application. And so just about to wrap up, I'll give you a quick view of the Shiny application that all of you can make pretty quickly. And so let's go ahead and look at all of the results here. And we're going to take a look at the position and build the same visualizations that we made before. And so let's take it for let's look at the position here. and we'll look at it or by the log 10. Here it is. And we're going to go ahead and do the facet or the groupings by the chromosome. Here we go. And so now you can see, here's the visualization that we just made. It's got all those interactive components that we talked about. But now, too, it also has the data. And so you can see these components over here are added in through the Shiny capabilities and are going to be a little bit more interactive and powerful compared to the HTML one that we just created. So hopefully this highlights the type of workflows that you can make. Just to recap for today, we went from a notebook to a dashboard to a Shiny app in 15 to 20 minutes. And all of this, I think, hopefully you can repeat and play around with. And I'll post these examples on the repository as our next speaker is talking. And hopefully, if you have any questions, feel free to put those in the chat box. All right, and Anastasia, yeah, we'll pass it over to you.
Right. Okay, so today I will be showing as a last presenter how you can actually use RStudio on the UKB wrap. So what I will show you actually is how you can launch RStudio Workbench, how you can work reproducibly in R using RNF library, and how you can save and restore RStudio projects from UKB wrap project storage. So I will be focusing on how you can actually work in RStudio within UKB wrap. So I'll just show you quickly the demo how we can launch RStudio, then we'll go quickly through the code, and then we'll look again back to RStudio Workbench, how it looks like on wrap. So here I am on UKB wrap. So as we saw from the post, so some of you are not part of a UKB wrap platform, the researchers yet. So this is how the UKB wrap platform looks like. And if I would like to launch RStudio, I will go to the tools section and you will see that there is RStudio Trial. And here, so if you will open it, you will not see these things because I launched RStudio many times. But you can configure RStudio session easily. So you click on New RStudio, you can configure environment name, or it will defaultly put a date and time. Then you need to select project. Selecting project is an essential thing because by selecting the project, you will actually mount it to the data, so the data from that project will be available. And because I will be showing GWAS visualization, so you need to select the project where the GWAS visualization output data will be available. For me, it's this. I'm selecting this project, and then there is a priority. We highly recommend you to let it as high because it will prevent from some unexpected turning off the instance, which will be really important when you're writing some code. And then you can select instance type. So here you can see that there are different instance type, and by looking on the icon, you will see what is the total memory, total storage, and how many cores it has. So, and by configuring that, you will also see the estimated price. And maybe for those of you who are used to Jupyter Lab, you will see the estimated time per session, and there is a default time set for session. But here for RStudio, we do not have it, meaning that here, so there are two important things. First is that you are seeing estimated price per hour, but the second, even more important thing is that you need to terminate your RStudio session once you're done. So I will not launch my RStudio right now because I have it already launched. So you will see that there will be initializing, launching, and once the RStudio instance will be ready, there will be an open link that you can open. So you can open it and you see that I have already a session here, but you will not see that because you will have the fresh RStudio. You click on new session, you let everything as default, and you can start new session. So this was very brief introduction how you can actually launch RStudio. Let's look back to the slide, and then we can again return to our RStudio Workbench. And the slide will be available just after the webinar. So you will be opening the RStudio Workbench, and the interface will be almost the same as you see the normal RStudio on your local machine. The only thing is this terminate button that you see here, and it will help you. It will remind you to terminate your RStudio instance once you are done with your work. So here I will be showing you how you can use REnv package for reproducible research. And this is a package manager for advanced R users. So I will be showing to you 2 ways how you can do that. And so the main workflow, if you are using REnv, is initialize REnv package manager, then install packages, and then make a snapshot. So the state of your R libraries is all written to your REnv log that can be then used once your RStudio project is restored. And we will be working inside RStudio projects. So we have the RAP project is the project of your main application. And then there's RStudio project, which is living inside RStudio. And we can save it back to the RAP project. So sorry for a little bit of confusion, the 2 projects we're talking about. And when we are talking about REnv package, in case this will be too complicated for you, you can start using your RStudio normally as you're using it in your local desktop. But then if you want to move to the more reproducible research, it's very beneficial to try to work with REnv package. So as I said, that the REnv package is titled with the RStudio project, and actually Phil was showing to you how you can use RStudio projects for reproducible work. So I can go just quickly through that. So you will create a new project, use REnv with that project, install it, and then you will see that the rproj file is created, which is your RStudio project, and also REnv log is created. And the log file will, after you will make a snapshot, as I mentioned before, will have all of the state of your R libraries that you installed inside your RStudio project. So here I'll just show you how you can download the GitHub repository content and then work with it and analyze some data. So in case you haven't seen any of our webinars before, we have our DNAnexus community GitHub, which is UKB_rep. And after the webinar, you will see the slides, so you can go and click on the links and look through the repository, because there is a lot of interesting code that we already published for RStudio and not only. So we have our GitHub repository inside our RStudio, and we will be working mainly with GWAS visualization, R Markdown, and I can show it to you here. So now I can go to the R Markdown here. So this is just to have a brief overview, so I jumped quickly to another window. So this is your RStudio inside UKB RAP. It's the same, as I said, almost the same, except for the terminate button here, you see. So we're going to, we have this GWAS visualization R Markdown, and we can go through the different steps to make a visualization. So we first need to, we will be working with the Regini output files. So I'll just show it to you quickly here. Yeah, sorry. So here is the file that we'll be working with, and Phil was already showing to you how you can do different visualization with that. I'll just make some technical note, the specifics of working on RAP. So we have some gzipped files for different chromosomes on our UKB RAP project. And in order to work with them inside our RStudio, we need either to download them using DX Toolkit, and I will talk about that in a moment, or the more convenient way is to use DX Fuse, and you can use it by adding to your file path, to the your Regini files, you're adding /mnt/project. So this allows you to use files from the corresponding UKB RAP project that we were selecting when we were launching RStudio Workbench instance. So by that, you're making some needed manipulation. So you're creating the file containing results for all chromosomes, because usually when you have output for regene, which is a GWAS2, you will have it per chromosome individually. Then you can read it inside R and then work with it as with the usual R data frame. So here we read the table, we rename the columns, and we separate columns. So this is our final data set, or like the final look of our data set after the cleaning. Then we can work with. And so then we can do any of the plotting, some of the plotting that Phil was showing, or any other plotting. So here I decided to make 2 plots: Manhattan plot with using QQman package, which looks like that, or interactive QQ plot with which looks like that. And I can show it to you moment in sorry. Yeah. So once we're done with our analysis and we want to actually store our work, including all of the libraries that we installed, we need to create a snapshot, and we do that using our RNV package. So we're creating a snapshot, and then in our RNV log file, we're having all of the states of the libraries that we had in our project, and then we need to transfer our work to the UKB RAP project. So remember, again, we have 2 different projects. We have UKB RAP project, which is permanent. It's on UKB RAP platform. And then we have another project, which is RStudio project inside RStudio instance, which is temporary, meaning that once you terminate your RStudio app instance, everything that was unsaved is gone. So that's why we need to establish the way how we can actually first move files from UKB RAP project, from the permanent projects, and then move them back, transfer the results to the UKB RAP project. So for the first step, we can use DX download, and I will talk about what is DX actually mean with DX toolkit in a moment. So either downloading some input files or downloading the whole project. And for the transferring, we can either again transfer the whole project or transfer only one file. So what is the DX toolkit that I already mentioned 2 times? This is DNAnexus developed a command line tool that can help you to work with files on UKB RAP. And there is a lot of utilities there, and you can go through documentation and see all of the list of the commands, and also look the videos that we created. But the important commands to us would be backup folder and DX restore folder to save and restore our studio projects. Also DX download to download file from project to the instance, to our studio when we want to work with files, and then using DX upload to upload our results from our studio back to the permanent storage. So these are the 4 main and important commands that we'll be using. So for us, once we are done with our work and we want to, we saved everything, we create a new log file with all of the libraries, we will use DX backup folder, and here we're specifying where we want to make a backup. And then in our permanent storage, in our project storage, we will see the gzip file with our RStudio project. Or if you don't want to work with RNV so far and you just want to try it the usual way, you can use DX upload and just upload, for example, your code, because we did not generate any output files. So you can use DX upload, specifying the file that we want to upload and specifying the path, and then you will see that the file will be uploaded into the permanent storage. So these are the 2 ways how you can save and upload your results back to the project. What if you decided to use the first part, the DX backup folder? You can then use the complementary function to that, which is DX restore folder, and then you can restore it back to your RStudio. So once, for example, on the next day or next week, you want to work with your data, you can use the DX restore folder, and then you can open your RStudio project and then work as usual. So by activating. And so once you're done with the whole your work, really do not forget to terminate RStudio Workbench, but do not forget to store your data before. Okay, so this was quick, and it was very quick introduction into how you can work with RStudio on RAP. If you're interested in more details, you can look into our webinar that we had in June on RStudio. And there will be some upcoming webinars. We'll have the analysis of accelerometer and metabolomic data from UK Biobank Dataset. It will be on September 15th, and there will be a Docker. And we already have more than 15 webinars already, so you can look to all webinars recording there. Okay, so, and we have, I think, about 2 minutes or 1 minute for Q&A.
Yes, thank you so much, Anastasia, and thank you so much, everybody, for all your great questions throughout the webinar. There's been a ton of great things coming across. I did want to mention about like RStudio right now on RAP. DNAnexus and UK Biobank recognize the importance of tooling like RStudio being available on the platform to support the widest range of users. So during this trial, we very much welcome user feedback during the remaining trial period. So we want to ensure that this feature remains accessible to users conducting research on RAP. So licensing and commercial options beyond the trial are currently under review. I know we got a question about that and people curious, so I wanted to mention that up top. So one that just came in was, I wasn't sure where you type DX download, like wanting to bring data from RAP into an R environment. Where exactly are you doing that to bring it into the R console?
Yeah, so for in RStudio console, like in RStudio Workbench, you see the different like RStudio console or terminal. So when you are using just DX download, you can put it into a terminal window, or as I was showing, as I'll have in the slides, if you want to use it inside your R script or R Markdown, you need to wrap it inside the system function. And system function is a way how you can call the terminal functions inside your R environment. So yeah, so how you can do that.
And I wanted to call another one that I thought was a good question that you answered, Anastasia, about querying phenotypic data within RStudio Workbench, like they do in Spark in Jupyter Lab.
Yeah, so we had RStudio webinar in June, and we created the R script where you can actually query the data, the phenotypic data inside RStudio console, and the data will be downloaded into your temporary instance. So we really advise you to look at our UKB_RAP GitHub repository, and then you can work and play with that with R script.
Perfect, thank you. And I know we're already over time, but I did want to ask one question to Alex that I also saw come in about UK Biobank data models. What data models are you using for observational data collection, e.g. OMOP, SNOMED, or others?
So this is totally not my field of expertise. So I've linked the documentation that was created when planning and executing this part of the research in the Biobank. And if you have more questions about it, please redirect it to our access team. They would be able to answer this in much more detail than I would be able to tell about it.
Perfect. Appreciate the clarification there, Alex. So we're just about out of time. Thank you so much for sticking for a few minutes after, and hope you enjoyed the webinar. Again, recording and slides and some of the helpful links that were dropped in the chat will be made available to you afterwards. So keep an eye on that within the next day or so, and on community@DNAnexus.com, where those will all be listed in a post. So I want to thank all of our speakers for joining today and running through this presentation, and for all of you for joining.
Thank you.